Tina, are you going to let people in? Good morning, those of you Advanced Kentucky AP Bio students, and welcome to our second AP Biology online content review session. Just very quickly, my name is Lou Akampora. It's 11 o'clock and we'll get started. You should be seeing my screen, and today Brittany Cox will be our presenter from Jessamine County High School. Brittany is a AP Biology reader and one of my go-to people for AP Bio questions. I am your host for science, and you may know Aaron, Tina, or Monique from other AP science courses. As we do this, please turn off your participant video and your microphones. Please do use the chat box. And if you've got a moment to just chat out where you're from, we'd appreciate that. This session and others are archived on the kyap2020.com site, and teachers may find other resources there, including notes from this session and others. So please go there if you have any questions. So before let me introduce Brittany. She had taught at Lincoln County High School, has been an AP Biology consultant for Advanced Kentucky for not many years. She's not young, not old like me, and is an AP Bio reader and brings a wealth of experience and expertise to this. So I will turn the controls over to her. Brittany?
Good morning. I hope you guys are all doing well this morning.
And you can go ahead and share your screen when you're ready, Ms. Cox.
How does that look?
Looks great now to me.
Okay. Well. All right, so today we are going to talk about DNA, central dogma, and I know that a few of us are probably still a little sleepy this morning. So I want to start with you guys to just have an opportunity to warm your brains up. So what I'd like you to do in the chat box is I'd like you to type anything that you can remember relating to DNA or the central dogma. Separate your thoughts with semicolons. And here's the hard part. Do not press enter until the timer sounds. And it's not going to let me start my timer. So I'll just tell you when time is up. You have a minute. It's really important that you guys participate with me today because I have probably more content than an hour will allow. So I'm going to determine pacing based on your guys' interaction. So if there are things that you already seem to know very well, then we will go quicker through that. And if not, then we will slow down and take a little more time with that content. All right, let's go ahead and pop those into the chat box.
So everyone should hit enter.
Great. DNA is a double helix. You read DNA 3 to 5. What else do we remember? Stored in the nucleus of eukaryotes, correct. Self-replicator. You guys are doing so great this morning. I really appreciate that participation. So here we are. And I think Lou is going to pop a poll up here for us so you can have some anonymity. But right now, I'd like you to take just a second and rate your confidence on molecular biology, DNA, and gene expression. Okay, so here's your rating scale. 1 is you don't feel very comfortable at all. And 5 is you're very sure that if you have an FRQ over this topic, you're going to do very well.
And we've got about half the participants, so don't think about this too much, folks. Whoa, who's choosing 5? That must be a teacher.
Ooh, above and beyond.
All right, I'm going to close the polling here in about 5, 4, 3, 2, and there we go. And looks like most people are pretty comfortable.
I'm really excited to hear that. We see that most of you are at a 3, which is a great place to be. So let's move along and see what you can tell me about this figure. So, sorry, the poll results popped up. What is this molecule that I'm pointing to up here at the top? What is this? In the chat box. Anyone remember what this molecule is? Okay, well, that's okay. Let's look at this picture that's already labeled. So this is a very simplified diagram of the central dogma that says all of our genetic code is contained within DNA. DNA is then transcribed into mRNA, and mRNA is then translated into polypeptides or proteins. So this is, you know, how life happens. So it's very, very important, and we're going to get to some of the details.
And those students who were here last week.
So we're going to do. Go ahead, Lou.
I'm sorry, I was saying those students who were here last week remember the importance of protein structure and how the primary, secondary, and tertiary and quaternary structures determine the function of a protein.
Yeah.
So this all ties together.
Definitely.
I'm sorry,
Yeah.
Brittany, I didn't mean to interrupt.
Oh, you're fine. So Lou is going to do a poll, and we're going to answer some of these multiple choice questions. So here's your first one.
While we're waiting for the poll results, Brittany, about how, when did Beadle and Tatum propose this? I'm old enough that the structure of DNA came out just before I came into this world. Do you know about what year Beadle and Tatum would have done this?
Oh, Lou, that's not a fair question.
I know you're a youngster. Okay, well, we've got a little bit more than half the participants voting. I'll give you about 10 more seconds to take your stab. And in 3, 2, 1, there's what we have. And can you all see that?
That's great. So the majority of our crew is choosing B, which is exactly what I want to see you guys doing. So let's talk about B and why it is the most correct choice. So as you guys worked with Mary Beth last week, we know that genes code for proteins and that lipids and carbohydrates come through a different way of synthesis. So you could eliminate C pretty easily. Now, when we look at A and B, those are the best two choices given there. And the reason B is better than A is that it indicates a given sequence of DNA. So a given sequence of DNA is what we are calling a gene, right? So B indicates that it's a gene, whereas A indicates that it's an entire DNA molecule. So B is the best answer to this question. All right, let's do it again. Lou, I think we're good if you want to go ahead and cut this one off. We had an overwhelming majority get this one right. Everyone answered A, which is great. Double-stranded DNA. Can DNA ever be not double-stranded? No. DNA is always double-stranded. And we know that it's DNA and not RNA because thymine is present instead of uracil. So good job, guys. One more question and we're going to do a little bit of content. Whoops. Good job, guys. It looks like we have the vast majority choosing answer choice A, which is correct, because the damaged bases have to be recognized before anything else can be done. Okay?
Ask her how long it takes for him to cook.
Kelly, usually no, it's not important that they know which scientist did it. It's just important that they're able to interpret the results. Sometimes the scientist names are included in the question stems, but usually it's not necessary to know the answer to the question. Does that make sense? Okay, so we're going to take just a second to look at the structure of DNA. And I'm pretty confident that you guys are going to be able to do this. So the monomer of DNA is a nucleotide. Does anyone remember the 3 parts of a nucleotide? What's this one that's got the 5 points? This is the pentose sugar, or the 5-carbon sugar. These are the nitrogenous bases, and these are the phosphate groups, okay? So I want to talk a little bit about Chargaff, and I think somebody mentioned this when we were sharing information at the beginning of the session. Good job, Amy. When we were sharing information at the beginning of the session, is that there are base pairing rules where we know that adenine is always going to pair with thymine and guanine is going to pair with cytosine, and that is was a discovery by Chargaff. So they may give you some percentage questions about that, but since the math section has gone by the wayside this year, I don't really anticipate it. Now, I think this is one of the most important parts that I see come up again and again, is you need to know about the different bond types. And yes, Lou, I know that the hydrogen bonds aren't real, but in biology, we say they're real. Okay, so these bonds that hold the sugar phosphate backbone together, those are covalent bonds because carbon is involved, right? These down here that are holding the 2 sides of the ladder together, those are hydrogen bonds, okay? So now we're going to talk a little bit about relative bond strength. Which one is stronger, a covalent bond or a hydrogen bond? Which one would take more energy? Oh, I love that. Look at that, Lou. They all know that the covalent bond is much stronger. Good job. So why do you think it's important that we have these super strong covalent bonds along the side of the sugar phosphate backbone? Sorry. Why do we want the covalent bonds over here? The covalent, yes. So we want the covalent strong bonds over here so the bond won't break. If these bonds on the side were breaking, then we would be losing portions of our DNA. And what would happen if we lost large portions of our DNA regularly? Nothing good, right? Be incompatible with life. Now these are hydrogen bonds. Yes, mutations. Good, Amy. These are hydrogen bonds in the middle. Why is it good to have a weaker bond here in the middle? You guys are doing a really good job. It wouldn't be the same protein produced if we lost pieces of DNA. Great, so these are weaker bonds because we do open the helix up regularly so that DNA can replicate, right? So these are weak so we can break them open, and these on the ends are very strong because we don't want to break them. Now the last tricky part, because you guys have not had organic chemistry, the prime numbers are a little confusing, right? It has to do with the number of carbon that the phosphate group is attaching to. So since I'm not going to teach you how to count carbons, I'm just going to tell you that whichever end has the exposed phosphate is the 5' end. So I just remember 5-phosphate, right? So if you need to find the directionality, look for the end that has this little phosphate group exposed. So there's why your functional groups matter from the beginning of the year. Does anyone have any major questions about this? Good. We'll keep going. So here, this picture is not very good, but couldn't find very good. These are the different models of replication that were proposed. We have, does anyone know which one is accurate, which one DNA replication follows? Is DNA replication semi-conservative, conservative, or dispersive? You guys are great. Very good job. Awesome. So semi-conservative, meaning that each new DNA has one side from the original DNA molecule and one side of newly synthesized DNA. And that is very good for us because it minimizes mutations, right? It minimizes mistakes. Good. So you guys are just killing it today. Kelly, are these your guys? They're doing such a good job. And Lou is going to post these slides. And like I said before, it's not important or necessary that you know the names of the scientists who did this. It just sometimes helps us organize our thinking when we think of the story of how DNA was figured out. All right. Henderson County kids, awesome. You guys are doing so good. All right. That's awesome. Good job, Morgan County. I'm proud of you too. All right. We're going to look at replication. We're going to focus. Yeah, that's what it was, Lou. We're going to look at replication. I'm going to focus primarily on the leading strand since this is just a recap, right? So I think the main thing about understanding replication is the relative bond strengths that we discussed before and the roles of these different enzymes. So I'm sure your teachers have told you over the last year that enzymes are special because they tell you what they do a lot of times. So helicase is an enzyme. How do we know that it's an enzyme? How do you know that that's an enzyme without me telling you that? ACE, good. It ends in ACE, so we know it's an enzyme. And what kind of molecule is it? Probably. It could be something else, but it's probably a protein. Good job, guys. You're on it. So helicase is an enzyme that opens the helix, right? So this is nice. You don't have to rely on memorization because if you look at the words, they tell you what they do. And that's one of my favorite test tips is if you start to panic, calm down, look at the words, see what they tell you themselves, okay? So helicase opens the helix, right? And the nitrogenous bases still have an affinity for one another, so that helix would like to seal back up. So the single-stranded binding proteins actually maintain the opening in the helix, so replication can take place. Topoisomerase, sometimes they talk about it, sometimes they don't. It's indicated here, and I like that they've showed it as a circle. It kind of rotates the helix as it opens to relieve the tension on the helix, to relieve the mechanical strain there, so that we do not damage the protein molecule. Primase creates a temporary RNA primer. See how it does that nice thing? Primase. Why do we need that primer? Does anyone know why we need primase? Why don't we just start making DNA? Yes, Kelly, good job. So DNA polymerase cannot start on its own. It needs something to bind to. So the primase comes in and lays an RNA primer, and then DNA polymerase can take off from there. And DNA polymerase, poly, I'm sure Mary Beth talked about this. Poly means multiple, right? Mer means parts. So the polymerase is adding many parts to this DNA molecule, and it adds nucleotides to the 3' end. It also has a proofreading ability, and I think it's pretty neat. It has to do with the length of the bonds in the center of the helix. So if things are not bonded correctly, they're not at the right height or position molecularly, talking very generally, right? And the DNA polymerase can recognize that there's an error that way, so that it can reduce the number of mistakes. And you guys talked before about how when we have mistakes in our DNA process, or we lose part of our DNA, that's a mutation, and mutations can change the protein that's going to be made. And it just depends on how significant the mutation is as to whether or not we'll lose function of that protein. So if we detect an error, nuclease, so again, it's an enzyme that's working on a nucleic acid. Nuclease excises the incorrect portion. What does excise mean? What does excise mean? Removes. Very good, Kaylin. Very good. Polymerase replaces with the correct nucleotide. So that's nothing new. Polymerase is sticking the nucleotides where they go. And ligase seals the excision points. Now, ligase is one that you guys may have trouble with because you don't know what that prefix lig means. But if you've seen the word ligand, what do ligands do or ligands do? What do they do with receptors? They bind. Very good. And do any of you watch true crime shows? Any SVU fans in the house? They also talk about ligature marks in shows like that, which are marks from being bound, right? So anytime you see that liga prefix, then you know that it is a bonding. So ligase binds that strand back together. So we add our nucleotides. Nuclease comes back in and clips out that RNA primer, because we're not making RNA, right? We're making DNA. And then DNA polymerase replaces with DNA nucleotides. The best way that I can tell you to study this, if you've not remembered it yet, is 1, flashcards with just the enzymes and their functions. 2, print a picture of the replication fork. Go through on the side and write the steps the best you can remember them, and then check them. If you can get to the point that you can write down all of the steps, then you're going to ace any question that has to do with this replication. Oops, sorry. The lagging strand undergoes the same process, right? Same enzymes, but the problem is it is not going into the replication fork. It is going away from the replication fork. So the main difference is it happens in these little fragments. So you have to create multiple RNA primers through the use of primates to do these little Okazaki fragments that are then sealed up with ligase. So you're seeing much more nuclease action, much more primase action, and we call it the lagging strand because obviously that's going to take longer than if we can just replicate continuously. And here are some extra resources that you guys can use. So if you're sitting here and you're thinking, wow, I don't remember any of that, then please take a moment to look at some of these resources, and that might help. Ready? We're going to do some questions. I think these are a little tougher than the last ones.
This one is a little bit tougher. A little bit more reading here. We're a little bit slower going, but take your time.
You guys are doing a really nice job. I'm feeling very confident for you guys come May.
And we'll give about 10 more seconds. Okay, 3, 2, 1, and there we go.
Wonderful. 74% of you chose answer choice A, which is correct. Oh no, sorry. It's the polling results. They keep throwing me off. Okay, so like I said, 74% of you chose answer choice A, which is the correct answer. Now, the reason that it is answer choice A, what made it kind of tricky, is you have to take into consideration the anti-parallel nature here. So the 5' end is going to, in fact, have to match up with the 3' end. And it's, you know, there you go. So you guys did a, wait. Yeah, you guys did a really good job. One second, guys. The polling results are stuck on the middle of my screen, so I can't see anything. Here we go. All right, next question.
Looks like we've got a pretty even distribution of choices right now.
Mm-hmm.
Okay, we've got about a minute here. Pass, I'll give you 10 more seconds. Three, two, one, and there we go.
Good. We have a slight majority here. The correct answer is D, because RNAi regulates in a pre-transcription manner. Okay, this is one of the old school questions that we've used at study sessions for at least the last 6 years. So it's an oldie but a goodie, but I always think it's kind of hard. And I really doubt that you guys would be asked something like this because it does rely a little bit more on recall. All right. This one is super tough based on the length of the question, I think.
This is a long question to read, and having a good reading skills will be very helpful. That reading load in AP Bio is pretty high. Okay, about 5 more seconds to make your choices. We've got about half of the participants voting. So 3, 2, 1, and
Good. So you're choosing C, which is the correct answer because it is the only one that happens after transcription. So good job. I want to take just a second. We're going to go back to this slide. I misread one of the answer choices trying to move the poll. I couldn't see it. So actually C is the better answer here because the tRNA is not carrying nucleotides. It is indeed carrying amino acids. So if you chose C, you are correct for this answer choice, okay? Sorry about that. I was telling Lou today that I'm pretty nervous about teaching online today. Okay. So we're gonna take a look at transcription. So just like we did before, I want you to take a few minutes to type into the chat box what you know about transcription, right? Or protein synthesis. And we're gonna wait just a minute or so before we press enter so everyone can see the answers all at once, okay? So go ahead, and then after just a minute or so, we'll ask you guys to press enter.
are we talking about transcription or translation here?
Transcription primarily, but if we can remember anything relevant about translation, I'll take that too.
Okay, sounds good.
So maybe about 10 more seconds. All right, let's submit your answers. Press enter, please.
Whoa.
They're doing great, Lou. I told you they'd use that chat box today.
I will say the bio students are much chattier than the physics students.
Yeah.
Good job, everyone.
You guys are doing such a good job. Good. All right, so protein synthesis is how genes are expressed. If a protein is not being synthesized, then that gene is in an off position. Okay, so this is a little tricky for students sometimes to make those one and the same in their minds, but a protein being expressed or a gene being expressed is in fact the protein being synthesized. So we're going to look at this first part of that process right now, where you're going from DNA to RNA, and this is transcription, right? So you're going from one nucleic acid to another nucleic acid. All right, so where does transcription occur in eukaryotes? Where does transcription occur in eukaryotes? Does anyone remember? Oh, there we go. Messages. Yeah, in the nucleus. And in the prokaryotes, it would happen in the cytoplasm. Today, we're going to focus on transcription in eukaryotes, due to a time constraint. But we got to remember our prokaryotic friends when you guys are preparing for the exam. So it happens in the nucleus, right? And we're making an mRNA primary transcript in eukaryotes. So you can break transcription into 3 different steps. And the first one is initiation, which makes perfect sense because initiation means to begin, okay? So RNA polymerase binds tightly to the promoter. What word do you see within the word promoter? What regular word do you see in there that you would not use in a science classroom? Promote. Good. So if I'm promoting something, what does that mean? Like if I'm promoting Coke over Pepsi, what does that mean? If I'm a Coke promoter, I'm endorsing it, right? I'm encouraging you to have some Coke. So the promoter, I think of it that same way, is that's what's getting it all started. It's saying, hey, come on, let's do the transcription right here. So RNA polymerase binds to the promoter. The promoter is letting RNA polymerase to know where to start and which strand of the DNA to use as the template. And when to transcribe or under which conditions. The transcription initiation complex assembles around the DNA. That looks really scary, but all it's telling you is that it's what starts the transcription, right? It's the pieces that work together to start transcription. Next is elongation, where your mRNA is going to be growing, right? And I'm just going through this to break down the words with you guys because I think often we're intimidated by some of the terminology, and especially on test day, we panic a little bit. Whereas if we feel like we can go in, figure out the words, break the question apart, it really helps us be successful in tackling those FRQ questions. Okay, so an elongation, the RNA polymerase exposes about 13 nucleotides at a time. It adds nucleosides to the 3' end. So the first nucleotide delayed will be at the 5' end. RNA polymerase proofreads too, but it proofreads with a lower degree of fidelity than the DNA polymerase does. What does that mean? If it's proofreading with a lower degree of fidelity than the DNA polymerase. It's not as what? Not as reliable, with less accuracy. Very good. Wonderful. So yeah, the RNA polymerase, it proofreads less accurately, which is less of a big deal because the mRNA transcripts are temporary, right? We're going to use them, then degrade them. Whereas if we make mistakes in the DNA, then we're going to have problems from that blueprint every time it is used for transcription. So the RNA polymerase, while it does proofread to minimize mutations, it does not proofread with the same degree of DNA polymerase. Oops. And we just talked about that. Finally, we have termination. What does termination mean? If I just got terminated, what does that mean? Deleted, end, fired. I was hoping for fired, but I can be deleted too. So termination means to end something, right? So during the termination step, transcription does end. Okay. I'm getting some interesting comments in the comment box over here. All right. Prior to termination, a stop codon is transcribed. Transcription slows down. And then the process terminates and mRNA transcript is released when the RNA polymerase forms AAUAA. All right. So I'm going to show you a very, very short video of this. It's like 2 minutes. Some of you are visual learners, so this is what you need to see it, right? Maybe. Here we go.
Transcription is the first step in the expression of a gene. It is the formation of a specific RNA sequence from a specific DNA sequence. Transcription initiation occurs at a promoter, a region on the DNA where RNA polymerase binds. RNA polymerase attaches to the promoter and begins to unwind the DNA. At the initiation site, the polymerase begins reading the DNA template strand and building a complementary RNA strand from free nucleoside triphosphates. The RNA strand grows by the addition of these substrate molecules to its 3' end. This is the elongation phase of transcription. Note that the bases in the nucleotides of the RNA strand form pairs with the bases in the DNA strand. Also, note that the RNA nucleotides contain the sugar ribose, which has a 2' hydroxyl group, whereas the DNA nucleotides contain the sugar deoxyribose, which lacks this hydroxyl group. RNA polymerase functions similarly to DNA polymerase. It adds nucleotides to the 3' hydroxyl group of the last nucleotide. The DNA double helix rewinds as the RNA polymerase moves through. When RNA polymerase reaches the termination site, the RNA transcript is released from the template. The DNA rewinds completely, and the RNA polymerase dissociates from it. The DNA and RNA polymerase can then participate in other rounds of transcription.
Okay, and there you have some more notes, just like we did for the last little subsection. Here's some other resources that you can use to review transcription if you are really struggling there. And I'm going to do just a little bit on post-transcription modification. This only happens in eukaryotes. And some of you mentioned this during the beginning when I asked you to post some things that you remembered about this process. So once the mRNA transcript is completed within the nucleus, it does undergo some modification. Why do we need modification before we send it out of the nucleus into the cytoplasm? Any ideas? What's it need to be protected from in the cytoplasm? Good, digestive enzymes, anything that could harm that mRNA because it would cause a mutation. So the first thing that happens is we get this methyl or guanine cap. It's added to the 5' end, okay? And that is gonna reduce damage in the cytoplasm. And it can also signal ribosome attachment. Then, for the same purpose, we attach a poly A tail, which is just a whole bunch of adenines tacked onto the end to protect from that digestion once again. And then finally, we have alternative splicing in the middle, and that's where we can remove the introns with spliceosomes. Spliceosomes are made of SNRPs, right? So these are all just molecular scissors. And then the remaining exons are expressed together. What's important here and why we call it alternative splicing is we can use, those are real words, Lou. They definitely are. We can have different series of exons together and therefore change the protein that's going to be synthesized. So this increases our genetic variety quite a bit. And this is the way that I like to think of it. So how many of you have made these paper snowflakes before? Throw a little signal up or say yes or whatever in the chat box if you've made these paper snowflakes. Yeah, right? We've all done this at some point. Some of us better than others. So everyone starts with the same sheet of paper. I like to think of that as my primary transcript. But then the character of our product or the snowflake is determined by what we choose to remove, right? So this is a very good analogy in my mind because I can look at my scissors as the spliceosomes and the SNRPs, removing the introns from my transcript, okay? All right. And then I have more pictures. Lou's telling me I'm almost out of time, so here we go. We're gonna do a few more questions. Sorry, guys. Okay, I'm gonna cut us off. We're struggling a little bit of this one, but the correct answer is C, right? We're looking for what is not a way that we increase our genetic variety, and we are not gonna change our DNA, right? In order to make some proteins. We keep the DNA the same. All right. So I'm just going to go through these last few slides quickly, right? Since we're out of time. So how would you describe translation outside of biology class, right? How would you describe translation outside of biology class? It's usually described as going from one form or one language to another. So I like to think of the nucleic acids as our first language, and then we're translating into amino acids or proteins. So if you get trippy on which step comes first, it's transcription because you're saying in the same language. And then the last step is translation because you are changing languages. Okay. Happens in the cytoplasm at the ribosome. mRNA is read in groups of 3 or codons. And tRNA carries free amino acids to the ribosomes. This is the genetic code. It's universal for the most part. Some prokaryotes get a little weird. It's redundant, and what that means is you can see 4 different codons in just this box are coding for serine. But it's not ambiguous. It doesn't mean that sometimes you put in UCU and you get a serine, and sometimes you put it in and you get a leucine. You always get serine from UCU, but there are different ways to get serine. And then the third thing I want to point out is wobble shift, which comes up from time to time. If you notice, this last position has more variation and it still gets serine. Like all of these start with UC, but then you see a multitude of nucleotides at the end, right? So this is because there's more likely to have a mutation in that third codon position, and they still code for the same thing. So they generally do not cause, those would be silent mutations, right? It would be a point mutation that was then silent because it did not change the amino acid or the structure of the protein. The ribosome is made of rRNA. The small subunit is the workspace. The large subunit does the actual assembling. There are 3 sites, okay? Key sites are where they're actually put together. So I think that's the most important kind of site. What type of bonds are being formed when the 2 amino acids are stuck together? They're covalent bonds, but they're more specifically, yeah, peptide bonds. You guys are doing great. Good job. It has 3 stages as well. And I'm just going to go through them. You have this slide. All right. Shoot, I've gone crazy. And then you have just some more review resources here. And I really encourage you to watch those AP lessons that are online because they can go into a lot more detail than we can here while we're trying to interact with one another. All right, nothing's to be feared, it's only to be understood. Now is the time to understand more so that we may fear less. Please adopt this philosophy as you prepare for the AP exam, and maybe as you just go from day to day right now. But the more we know about the exam and the more we study, the less anxious we'll be as it approaches. And I think that's a lot of it, is just being confident heading into exam day. I want to encourage you guys to reach out to me. I always look forward to this time of year, and it's sad that I won't get to see you in person, and especially some of my colleagues. This is the only time I see you. So reach out if there's anything I can do. Finally, don't touch your face, and please wash your hands. From a biological perspective, stay safe. All right, thank you guys.
And thank you so much, Brittany, and thank you all the students. You are terrific. This is more participation than I've ever seen in a science chat, but you are wonderful. I learned a lot of biology. This is a difficult time for everyone, but it's also a time of opportunity, and I hope you are really enjoying learning. This is a fantastic class. The video, the archive will be available, and contact Brittany with any questions about biology or me with any other questions about chemistry or science. And stay safe. We care about you guys. Bye-bye now.